Hello, this is Neil O'Shea, Managing Director of Discern Sustainability, a sustainable and responsible investment consultancy. And this is the first in a series of occasional podcasts. Imagine, if you will, an hermetically sealed vat full of strange organisms. Strange because they can perform apparent miracles by design that evolution has not. Stranger because, while not quite alien, they bear only a passing genetic resemblance to naturally evolved progenitors. Not even speaking the same genetic language, though they share the same alphabet, they are consequently sovereign entities, neither vulnerable nor threatening, in splendid isolation, parallel but never touching, except under controlled conditions deployed for their intended functions. Now imagine a century's worth of data compressed into material that would fit in one warehouse. If I asked you what could enable these two disparate visions, and if you were to say synthetic biology, I would say that you are a very astute reader of the title of this presentation. Now, on a serious note, listener discretion is advised with the topic before us. It does deal with a taboo and is likely to provoke strong reactions. For what it is worth, I am still agnostic on whether the brave new world Synbio heralds is to be welcomed, for reasons that will become starkly clear by the end of this presentation. I am also at the limits of my own ignorance on this topic. There are doubtless simplifications in what I present that would cause biologists or even engineers some grief. But part of what motivated me to prepare this introduction to this topic is that the words synthetic biology draw blank expressions from people in my peer group. It is too important to pick something easier. So what is synthetic biology? Most of us are familiar with the relatively established practice of gene editing. People tend to think of genetically modified organisms, GMOs, first, and yes, GMOs are examples of edits to what is already there. This may be as small as silencing a gene responsible for a disease, or adding one that will give the organism new properties. Synthetic biology, on the other hand, shifts gene editing from adjustments post hoc to fundamental design ex ante. In its purest expression, Synbio is the writing and programming of the all-ware of life, DNA. Its purpose is to instruct proteins and enzymes to carry out novel and useful functions. Proteins are the makers, the army of life, and the genome is its rulebook. A side note, but speaking of simplifications, the popular science of Synbio is rife with the shorthand of software and hardware, software standing in for genetic coding and hardware for the messy stuff that does the work. My reference a moment ago to all-ware reflects the reality that there is no such clear distinction in living cells, making them more recalcitrant and difficult to direct. A hyperjourney of gene editing sees it start with the agricultural revolution around 10,000 years ago, when humans first began to cross strains. They were gene editing, they just didn't know the molecular mechanics of the how and why. Crossing seeds, creating new strains, was crude and progressed slowly through trial and error. The 1990s marked the era of GM, which included the transgene method of splicing in genes from other species or environments, something that remains controversial, though in my view, unjustifiably so. We are now entering the era of directed evolution, where we are starting to engineer de novo molecules starting with an empty petri dish. What has catalyzed this first principles bottom-up approach are the discovery of accurate, precise and accessible cut-and-paste RNA/DNA tools, the advent of high-throughput genome screening by computers smashing through Moore's law, and an artificial intelligence finding, modelling and reproducing hidden connections and functions between genetic material and proteins. The field is still in its infancy, but it is showing signs of prodigy. It is a technology, not an industry, and I should manage expectations about how far we have got towards creating 100% artificial life in the meaningful sense of the term. As of today, we have theoretically viable 100% synthetic cells, but they are far from being an autonomous organism. The cells don't pass all the established tests for life, including the ability to mutate and reproduce. For example, in 2016, the godfather and maverick of gene editing, Craig Venter, stripped down a cell of its genetic contents to the bare minimum needed to operate, while in 2018, the other most charismatic character, George Church of Harvard, went the other way and built up such a cell. Let's spend a little longer on the simplified mechanics of gene editing in the synbio age. Proteins work by catalyzing chemical reactions that build into useful functions, a hormone or a pesticide, for instance. Research suggests that the humble yeast is malleable enough to be developed into most proteins. Gene editing downstream of DNA manufacture requires locating exactly the genetic material whose code needs changing, silencing, or removing. Gene editing can perform changes without exogenous DNA, for example, a knockdown of an undesirable gene, or go all the way to full synthesis. This is manufacturing function in life, if not yet life itself. Success in synbio depends on the convergence of the sourcing of base chemicals and their combining to print DNA. This makes it sound simple, and at least at the beginning, it is, allegedly. Within 5 minutes of finishing this presentation, you can Google and order to your house the base chemicals of genes, the codons of A, T, G, and C. These are usually derived from sugarcane and come in powder form. Now, assuming you knew how to code the letters in the correct sequence, they must then be assembled and stitched together, up and up, by means of robotic pipetting, into the right combination of amino acids from a family of 20. The challenge then becomes a lot harder. You have to get the amino acids to fold up into the scaffold or polymer-like shape needed to form an operable protein. Further still, there are the neighbours to consider. Can you get your new protein to play nicely with existing ones? As such, synbio is an inversion of top-down to bottom-up. The result is articulating new chemical instructions on virgin genetic pathways. Going back to yeast, its genome has now been entirely rewritten, but by rewritten I do not mean effaced. We will always have the naturally evolved ones, and this fact tethers us to the anchoring reality that synbio is less a binary state than it is a process, a question of variation and degree. This is why it was necessary to include gene editing in the title of this presentation. Gene editing, relatively modest, and synbio, as radical as it comes, cannot be separated. The celebrity in gene editing is CRISPR-Cas9. Discovered by Dr. Jennifer Doudna and Emmanuel Charpentier in 2012, it is a molecule combining RNA, that's the CRISPR bit, and an enzyme called Cas9. CRISPR acts like a sat-nav, homing in on exactly the strand of DNA that needs changing. The Cas9 enzyme that hitched a ride then acts as a surgical scissors to snip out and, if necessary, insert new material. If successful, the DNA strand repairs itself. There is no requirement for transgenes, in other words, genes from other species, but the system can accommodate it. Why it is such a watershed moment is because CRISPR combines those qualities that make it prolific. It is elegant, accurate, accessible, and cheap. There are middle schools in the US that are using it today. CRISPR, because of its genolocation knack, can be used solely for screening and detection, or for genetic surgery. CRISPR is not the only show in town, but for the present it is the best. Other methods are under development. Currently, there are more than 2,000 gene editing medical trials underway, mostly in the US and China. And most are still far away from commercialization, though the first trial participants have been treated. A joke you see going around is that synthetic biology consists of biologists pretending to be engineers and engineers pretending to be biologists. It's not a very good joke, but the point is that the objectives of the discipline are to, as far as possible, to identify, engineer, and standardize the parts that build the engine of life and its functions. Like Lego, they were dubbed by one leading figure as biobricks. Another objective is to reverse what is known as Eroom's law, or Moore's law in reverse, that regrettable phenomenon in big pharma in the last decade of ever more research and development spend producing even fewer blockbuster drugs. And finally, the objective of generating utility at scale. And scale is the operative word here, as we will see later on. And just look at that graph of what has happened to the cost of sequencing DNA. The first genome ever sequenced is estimated to have cost $1 billion. Presently, it costs about $1,000 and is soon to reach $100, putting the plummeting cost of solar in the shade. When I talked about that warehouse containing a century's worth of data, it is through writing digital code on strands of DNA that this could be achieved, a far more dense and long-lived form of storage than anything today. The current barrier is more of cost than feasibility. Predictably, it has been brute computing power that has put us on a virtuous learning cycle, writing out genomes as digital code. Cells and protein expression have been shown to display analog and digital computing and functionality. It is a rules-based system, hence my play on Corbusier's phrase for his high-density housing, machines for living. This high-throughput approach combined with artificial intelligence is leading to greater learning, efficiency, and lower costs, making each cycle more productive than the last. Examples of gene-edited and semi-synthetic products that are extant are chemicals, raw materials for plastics, food additives, fragrances, and biofuels. These are already being churned out at scale by altered microorganisms in fermentation tanks. As far back as 1980, the first patent was granted for a microbe engineered to absorb oil, an anti-pollution agent. You may already know that most of the world's supply of insulin is synthetic. Recombinant biosynthetic human insulin was first commercialized in 1983. The best-known antimalarial molecule comes from the Chinese wormwood plant, its discovery finally winning 2UU the Nobel Prize. The difficulty in harvesting it at first led to the creation of a synthetic version from industrial yeast. Perhaps the most eye-popping example of commercial editing is the Aqua Advantage salmon. 2 additional genes are taken from other varieties, and the result is that adults gain weight at twice the rate of regular farmed salmon, with 20% lower inputs. To allay biosafety concerns, the fish are sterile and only licensed to be farmed inland. All therapies being investigated in the US involve editing somatic or non-reproductive cells. Alterations will not cascade into future generations. In terms of the market's worth, Deutsche Bank in April of 2019 estimated the global market for synthetic biology, the bottom-up kind in other words, to be worth $40 billion by 2020. Investment in SynBio grew by a factor of 4 from 2015 to 2019. Synthetic biology and gene editing's full potential is best explored via means of a system, in this case agriculture, where its utility starts to look pervasive when paired with other technologies. Cows could be bred without horns, so that they need never go through the painful dehorning process. Nor would male chicks have to be grinded into a pulp the day they are born, because embryos can be edited to produce the female chicks, which are the economically useful ones. Going back to cows again, we're familiar now with the fact that cows' burps and farts are a huge contributor to global warming methane emissions. The effort to adapt cows' digestive systems to be more efficient and less emitting could be fast-tracked through gene editing of the animal and the genetic modification of animal feed. But what about the nasty stuff that still comes out the other end? It too could be made more productive and less polluting by means of edited microbes that could improve manure and biogas quality and efficiency. More precise animal genetics combined with better diet can increase meat and dairy yield for the same or lower inputs, like the AquAdvantage salmon. SynBio is already working on breaking pathways for trans-species diseases like swine flu. For plant-based agriculture here on the right, almost all agrochemicals, fertilizers, pesticides and others come from fossil fuels, not to mention their non-climate related side effects such as eutrophication, impact on pollinators and so on. While SynBio chemicals would hardly be free of unwanted impacts, built from the ground up rather than retro-engineered, their footprint should be more directed and manageable. Continuing with the plant theme, soil-enriching edited microbes would speed up cultivability in unproductive, marginal land unsuitable for forestry. Crops could be given inbuilt disease resistance, especially for those that are systemically important or relied upon by the vulnerable. Looking below ground, crop science companies continue to explore genetic editing for enhancing the root system for plants so that they sequester more carbon or fix more nitrogen. These traits can live within a network of the Internet of Things, where biosensors, microdata about changing environmental conditions, and the use of no-till farming allow accurate, dynamic, and resource-efficient dosing and crop rotation. For decades, crop scientists have explored the metabolic systems of plants, including the miracle of photosynthesis, to seek out improvements in productivity and resilience. Gene editing companies are working on increasing resilience to drought, flooding, and saline soil in anticipation of the ravages of climate change. I think it's vital to emphasize in all this breathless thrall to bleeding-edge science that work of this nature has actually been going on for decades. It is not theoretical, it's just that progress has been slow, at least in part because we did not have the tools and knowledge that are now within our grasp. Even more important is to appreciate that these techniques are not mutually exclusive with traditional, what some would call more biorhythmic, conventionally sustainable forms of agriculture. Although we don't have time to get into it here, organic and GM, as they are commonly misunderstood, is not an either-or situation. Not only can they cohabit, they can intermix. No-till farming and crop rotation were the norm thousands of years ago. It's just that they have a new role within a family of techniques aimed at working with nature's potential within the genomic rulebook. So here's what gene editing and SynBio don't fix, or could help exacerbate in agriculture. They don't address the risks of extreme proximity of animals and the denial of natural stimulation and instincts. Something you will encounter again and again is that there is no free lunch. There are hidden resource burdens and new externalities, the unintended or overlooked consequences. An analogy being the problems surrounding biofuels and their life cycle analysis, with potential for displacement of food, deforestation, and so on. We should not be so naive as to think that SynBio could not be used for cynical purposes to maximize profit at the even greater expense of animals or the environment. It would be more realistic to imagine that SynBio fits with an increasing trend of agricultural intensification. Indeed, sustainable intensification is the chimera we are after if we are to feed the 10 billion people Earth may be hosting by 2050. But gene editing's capabilities add to moral hazard. The fault with that lies with humans, not technology. SynBio also does little to address the rate of declining biodiversity. Of course, there will also be what are referred to as off-target effects. In plant science, modifications to alter a plant's architecture in one way, while successful, often lead to defects in another. Now, this phenomenon is observable in all areas of endeavour, but the fact that life is by nature ineluctable, intractable, sometimes unbiddable, means that off-target effects are even less visible. There is too the base fact that increased systemic complexity raises the margin of error and obscures the causes and pathologies of problems. Where we are closest to breakthrough technologies, and the ones you will read about on the news, are in fresh ways to tackle diseases which elude and resist conventional treatments. CRISPR isn't only for gene editing. Its identification prowess can be used just for screening for diseases. For example, companies Mammoth and Mission Bio are screening for multiple mutations within a single cell. The co-presence of disease indicators could result in a very different treatment regime. Companies like Zymergen and Microsoft Station B are scanning molecules for utility and modeling new applications via editing to repurpose for new applications. While this is of course the bread and butter of biotechnology, these supercomputer platforms are becoming a match for the palette of opportunities we can glimpse on the horizon. Gene editing allows us to create vats or foundries of edited cells, including those that have been taken from the body and edited before being reinserted as part of immunotherapy. Though there are no gene-edited approved treatments yet, we are close. A person has been treated for blood disease using CRISPR. The conjunction of gene editing and 3D printing is seeking to replicate or even improve upon materials in the body that become damaged, such as super strong spider silk filament that is aimed for use to replace damaged ligaments, once again derived from edited yeast. Gene editing, 3D printing, and nanomaterials could be used to model and construct tiny scaffolds to enhance cell adherence, lowering the chance of rejection in the body after organ transplants. Most investors think of drugs as the obvious sources of revenues from biotechnology today, but this graph shows that upstream biotech raw materials, for example, plastics, food additives, and other hidden applications are worth more. Productivity here in terms of useful discoveries is huge, though the ones that make money tend to be highly specific to the incremental and sometimes quite banal needs of its industrial clients, synthesizing a desirable aroma, reducing spoilage, and so on. The productivity comes from the brute force of computers and mass spectrometers gorging on, analyzing, and running experiments on data. For pharma, SynBio offers the potential to not just reverse out of an R&D cul-de-sac, but leap over the chicane into the biological superhighway. A tipping point has passed where this self-correcting model is more efficient than a human-centered model, though it is by no means autonomous. Ginkgo Bioworks, a leading gene editing company, went from a painful learning experience of being far less productive and more error-prone when using AI and machine learning to the opposite. They have passed that pain point. Human input is now more supervisory and interpretive in nature. AI modeling can, at its best, overcome limits of human observation when designing experiments, improving the signal-to-noise ratio from results, and thus make replicability much more common and rapid. This is what you could call BioWare-based innovation as a service. Let's take a look at some of the most well-known companies full of biologists trying to be engineers and engineers trying to be biologists. Ginkgo Bioworks focuses on two areas. The first is cultured ingredients such as perfumes like synthesized rose, food and cosmetics. The second is strain improvements, yeast and enzyme design to enhance efficiency and yield in industrial fermentation, for example, dairy, pharma and apparel. They have a joint venture with Bayer called Join Bio, scanning soil microbes to find ones that could turbocharge nitrogen fixing from the atmosphere. Its business, Motif Ingredients, aims to reverse engineer sensory experience of animal products to find underlying proteins and synthesize them. Do bacon first, please. At the time of writing, Ginkgo has raised $429 million. It is unlisted, but its purchase of company Synlogic stock caused the latter's share price to rise 22%. Ginkgo are focused on upstream services. Codexis is the second largest publicly listed, with a market cap of around 800 million. In its words, Codexis CodeEvolver platform combines DNA library synthesis and sequencing, advanced high-throughput screening, and machine learning with human expertise. End products are proteins that have been mutated by design to deliver desired functions for pharma companies. Codexis licensed the CodeEvolver platform to GSK in 2004 and to Merck in 2015 for development of novel enzymes, and to Nestlé Health Science for the development of novel biotherapeutics. Nestlé is targeting a condition called PKU and other metabolic disorders. Intrexon styles itself as the original synthetic biology company. Kudos for grabbing that website and domain, www.dna.com. The largest are publicly listed with a 1 billion market cap. Its piloted products include clean-burning isobutanol, a drop-in organic compound blendable with gasoline, and butanediol, a precursor to synthetic rubber. In healthcare, its phase 2 trials include biopharma molecules to treat oral mucositis. Another molecule, FCX007, targets fatal genetic disease, RDEB, through autologous dermal fibroblasts, genetically modified to express functional COL7, me neither, which is missing or deficient. Intrexon sold a controlling share in AquaBounty, the AquaAdvantage salmon, and now sells gene-edited, non-browning fruit direct to retailers. It is also responsible for the controversial friendly 80s mosquitoes that have next-generation sterility built in. The idea is to purposefully cascade the defect through the malaria-bearing mosquito population, causing periodic collapse. Unlike Ginkgo, Intrexon is focused on downstream products. Finally, Zymergen leans more towards the AI analytics side. It describes itself as a proprietary platform which combines advances in artificial intelligence, robotic lab automation, and cutting-edge genomics to unlock previously inaccessible sources of molecular diversity based on sustainable and renewable resources. Nice elevator pitch. Zymergen has built a vast vault of DNA to analyze. It is focused on bio-based substitutes for fossil-constrained specialty chemicals and electronics, for example smartphones, working with yeast and microbes as raw material. It launched a partnership with Sumitomo Chemical in April 2019. It is similar to Ginkgo in restricting itself to improvements of highly specific industry upstream needs. This table gives you a sense of how Big Pharma is approaching gene editing and synthetic biology. But it could be summed up as suck it and see at a safe distance. There's a reason why you've almost certainly never heard of the companies on this tree unless you are steeped in the world of biotech. The area has all the classic symptoms of biotech: young, bold, growing, and prone to failure more than success. It is a reasonable bet that most of these companies won't exist in 10 years. But that may not matter if the trial by fire they have gone through, having incinerated many, has left over a body of knowledge and know-how that can be taken forward by those with bigger balance sheets and more business sense. Alternative ways to access or just spectate on SynBio and gene editing are to large biotech companies who collaborate or absorb the pure plays, high-throughput screening companies that will be familiar to you like Danaher and Thermo Fisher, and of course the tentacles of big tech. The first reason to be skeptical about all this is that truly life is hard. One SynBio authority likens the process to disassembling a car and putting it back together again. Even mechanics would make mistakes, leave things on the floor. Life is ineluctable, mysterious, intransigent, and fiendishly complicated. Then there are the known unknowns. We know that alterations will have unintended and sometimes deleterious off-target effects. They can introduce polluting DNA, whose existence, never mind whereabouts or impacts, may not surface for a long time. Scaling up is a huge practical problem. There is no free lunch, no new matter or energy to create, just what is already there that we can redeploy. Robbing Peter to pay Paul. A great example of this, one attempt to create biofuels from algae. The company was able to increase oil productivity per unit of algae by starving it of nitrogen, but that suffocates growth. They then identified and edited the genetic switch that allowed the algae to multiply in the presence of lots of nitrogen. They got an improvement of 40%, but that is not yet enough to compete with the energy density of fossil oil, which has had the benefit of millions of years of geological and chemical processes. Despite the apparent bravado of SynBio, we still don't actually know the genetics that underlie human traits. So even if a clinic could claim to edit for intelligence, etc., they would almost certainly get it wrong. I talked about computers taking over from humans on conducting experiments, but it would be easy to overlook the lack of rigor being applied to spurious correlations inevitably found by AI techniques. It's been observed in other fields that AI reflects even the unconscious biases of its supervisors. BioWare-based innovation as a service spells bias. The second reason to be skeptical is a more mundane one. The challenge of making it through the tech valley of death made infamous in the dot-com bubble. Not only must solutions have a problem to look for, but existing solutions can remain stubbornly cheap, if unsustainable, such as oil versus biofuels. Even synthetic artemisinin, created as a public good, fell victim to private forces. Artemisinin found in its natural state fell in price, dislodging the synthetic stuff. Executives say that the problem is one of too many opportunities and lack of precedent to know where to most profitably focus. The artemisinin example shows us that just because something is possible doesn't mean it will justify its place in the industrial ecosystem. The sector is also distinctive in being both highly collaborative and acquisitive. Companies come and go. Urshit Parikh, partner at Mayfield VC, makes the observation that the typical mistake of disruptive hopefuls is to burn too much cash building the amazing product, leaving little in the kitty to build the market and service debt. They fail to pay attention to a working business model that can satisfy the precise needs and constraints of their clients. It is instructive here that the two most successful of the companies that we looked at by relevant investment metrics for this sector at this stage are Ginkgo and Zymergen, both solving practical problems of the here and now as directed by their clients. However, my reasons for skepticism should not be seen as pouring cold water over the area. I am just acknowledging known challenges that may be overcome quicker than we think, thanks to exponential improvements in technology and falling cost curves. Yesterday's problems will not always be tomorrow's. In the US, in-species gene editing sits outside the already permissive regime for genetically modified organisms. The reason for this is that existing biosafety protocols are deemed adequate. There are some grounds for this. Going back to the example of the not quite natural, not quite alien organism I hypothesized at the beginning, the reality is that industrial de novo proteins or bacteria are dependent on lab conditions for what limited functionality they have. They are captive, they are hobbled by design. The EU, on the other hand, is defaulting to its existing crypto exclusion regime for GM, whether transgene or in specie. Now hear this, gene editing in humans is widely, effectively, universally prohibited, including, for example, by the Council of Europe's 1997 Convention on Human Rights and Biomedicine. In the UK and US, genetically altered embryos must be destroyed before developing and never implanted, except in rare cases of mitochondrial disease, where the benefits will still not be heritable. In the US, no tax dollars may go to editing embryos of any kind, for example stem cell research, but the private sector is unfettered and it is going full steam ahead. This still leaves a perilous regulatory vacuum. An article in the journal Nature from 18 specialists in 7 countries called for a moratorium on clinical use of gene editing techniques for eggs, sperm and embryos until we have an international framework. Some prominent scientists, like CRISPR inventor Jennifer Doudna, demurred, saying this was kicking the can down the road and there would be no means of enforcement. Doudna's challenge is whether we should extend moratoria as we go, or should we just get on with the hard work of regulation, which could mean gene editing in humans is banned until it is regulated. What do the authorities in bioethics say? The UK Nuffield Council in 2018 pronounced that editing of human genome is morally permissible if it improves the welfare of the person without conferring anything that would disadvantage others or increase discrimination or division in society. End quote. You can see how a coach and horses could be driven through that superficially plausible sounding maxim. The Nuffield position also creates a catch-22 fudge. There are no examples of a person who's edited heritable DNA to know for sure that they have not been harmed, and no attempts should proceed until there is such a DNA edited person. The US National Academy of Sciences in 2017 said something similar, that human genome editing was permissible to alleviate suffering so long as no advantage, such as intelligence or looks, were conferred. Nuffield went on to concede that there was no clear distinction between disease prevention and enhancement. An example given of the imperfect distinction between curing disease and conferring advantage would be improvements to metabolism as a consequence of trying to avoid congenital type 1 diabetes. As for industry, trying to get ahead of the brewing malaise, it recently said it was going to make a declaration of voluntary rules its signatories would follow. The article in the Financial Times here on the left included language from the statement like, "The following activities are not currently appropriate." Genetically edited weasel words. Although the idea of edits cascading through the generations can be thrilling and terrifying to behold, the reality is that heritable changes will, only over a very, very long time, result in permanent changes to the human genome. Cybernetic changes and the so-called singularity, where human and artificial intelligence merge, are more likely to achieve the deification that Yuval Noah Harari calls Homo Deus first. However, the Rubicon of cascading edits has already been crossed. In November 2018, Dr. He Lianqiu announces from a lab in China that he has used CRISPR to edit the genes of 2 twin girls before birth to knock down the CCR5 mutation inherited from their HIV-positive father. He outrages the scientific community, including China's, breaking the cardinal rule of never engineering traits that are heritable. It later emerges that knocking down CCR5 may also significantly increase the chances of lower lifespan. It will surprise no one that Dr. He was soon after disappeared. In the days and weeks following this bombshell, it was not clear, even to Chinese officials, whether his actions were, strictly speaking, illegal or just incompatible with the intent of laws not conceived for this event. So, I've deliberately postponed acknowledging the tremendous ethical ferment that the future of gene editing will plunge us into, because I wanted us to get a handle first on what is and what isn't happening. Now let's dip into, if not debate, the arguments of those against full-scale gene editing and then those in favour. Those against raise the spectre of eugenics, of course, and all the old but compelling arguments about utopia really being dystopia, that we could witness speciation by income, as those with wealth have the power to upgrade themselves and their progeny to the detriment of the rest of us. We can take it as inevitable that some individuals, companies, and nation states will go rogue. Good and bad actors will create intended and unintended consequences. Your genetic data and what it supposedly says about you, your family, your likely behaviour, predilections, predispositions, lifespan, etc. will be the new frontier for exploitation. This is too much temptation for the abuse of authority. Who gets to decide what is permissible, using what criteria? The genetic edits, especially if they cascade, will be untraceable and irrevocable. Experiments in China admitted they have already lost track of participants. One can foresee a breakdown in trust and in the social fabric, a world more structurally determined, even less egalitarian. Could we be inviting an erosion of randomness and increased determinism, dispensing with humanist values? Yielding to a grim totalitarian rationalism married to capitalism, red in tooth and claw? What's the catch, I hear you ask? Okay, now in fairness, I also have to make a strong case in favour. And those in favour say that we should secure the burning platform that is our planet to allow future generations the chance to set their own norms by just assuring their existence in the first place. Who are we to deny tools that can increase prosperity, mitigate harm, and alleviate suffering? As for fears of being overrun by GMO swamp creatures, proponents point to the fact that lab-created synthetic life would be hugely disadvantaged in the wild against evolved and adapted genes. The pragmatists will say, it's going to happen anyway, let's get out in front of it. The optimists say that sustainable synbio, done right, should be mimetic of the best of biology to stay within planetary boundaries. A pedantic but important qualification from an existential or even theological perspective. Even 100% pure symbio is a misnomer, a contradiction in terms even. It is not strictly creation. We are still bound by what the fundamental laws and limits of physics and biology say. I'm not able to play this video at the moment, but it contains a range of very, very interesting and esteemed characters from the world of synthetic biology. And that man there, Drew Endy, is the one who used that analogy of trying to put back together a car again once you've dismantled it. Even the experts would find it bamboozling. He is one of the most eloquent and thoughtful authorities on this topic, and he tends towards optimism. They're on stage with an audience, presumably of mostly scientists, being interviewed by a journalist, and what is striking in this 90-minute long video is that the journalist takes a poll from the audience on the question of should we go ahead with engineering humans with synthetic genomes. I have to tell you that a large majority of hands went up. Indeed, you could not escape the whiff of hubris and ego among the panel while watching this video. Scientists doing what was deemed to be impossible because they can. Climbing Everest, in JFK's words, because it is there. Doing it because it is hard, not because it is easy. Some of you will remember a movie called Jurassic Park, in which there was a certain unpleasantness with some reanimated dinosaurs. It gave rise to some classic lines like, "Life finds a way," and "Your scientists were so preoccupied with whether or not they could that they didn't stop to think if they should." Yes, ladies and gentlemen, I fear if we are not careful and this technology gets into the wrong hands, we could usher in the return of Jeff Goldblum. Now, I like Jeff really, I'm just a bit jealous of him. Of course, artificial intelligence is the parallel our minds run to for whether Synbio promises a cornucopia or an existential threat, or both. Considering the fuss the tinkering of GM left behind, Synbio could be the mother and father of all taboos. As I said at the beginning of the presentation, it is not fence-sitting to say that there are myriad shades of incompatible yet legitimate views on this subject, rightly driven by hearts and minds. In any case, for or against, humans have a lousy record at predicting the future. It might be the single greatest existential choice we as a species ever make, except it's not really a choice because there is no controlling mind making it. What makes us think we will have more foresight this time around than we did with the internet? It will just happen in some form, and we will have to adapt as best we can, as we have always done. I'm going to leave you with some questions that we are working on for those considering investing in Synbio. It is too early to have all the answers nailed down, but people who care about the future need to confront the exhilarating and daunting possibilities hoving into view. We would love to hear your thoughts on all of this, so please get in touch at niall@discernsustainability.com. I also urge you to read Oliver Morton's magisterial treatment of this topic in the Technology Quarterly of The Economist in April of 2019, to which this podcast owes a debt. This is Neil O'Shea saying goodbye until next time, or should that be, go forth and multiply.